Electrophysiological recordings from muscle and motor neurons can be obtained from a properly prepared Drosophila embryo. Body wall muscles 6, 7, 12, or 13 can be easily recorded from the whole cell recording configuration, and robust miniature amplitude currents can be measured after achieving a gigaohm seal. To record from the neuromuscular junction, a suction electrode can be used to draw up a motor nerve near the CNS exit point and deliver presynaptic stimulation while whole cell currents are measured in the postsynaptic muscle. Alternatively, a pipette may be used to deliver excitatory neurotransmitters near the recording electrode via iontophoresis or pressure ejection.
Hi, I'm Kai-Yun Chen from the lab of Dave Featherstone in the Department of Biological Sciences at the University of Illinois at Chicago. Today, I will show you electrophysiologically recording the Drosophila late-stage embryonic neuromuscular junction and central neurons. These procedures we're using to study the function and development of Drosophila neuromuscular system. These techniques were originally developed by Kendall Brody, passed through down to David Featherstone, and then to me. Today, I will pass it on to you.
This procedure requires that embryos be properly harvested and dissected in advance. To see how this is accomplished, view the accompanying Jove publication by Brody, Featherstone, and Chen. Electrophysiological recordings from Drosophila embryonic muscles and neurons utilizes standard whole cell patch clamping techniques. First, the dissected embryo is placed in a small plexiglass recording chamber. The physiology recording area is electrically dampened by a Faraday cage and stationed on a vibrationally dampened table. Chamber is viewed under transmitted light using an upright compound microscope with Nomarski optics and a 40x long working distance water immersion objective. Recordings are typically made below room temperature, as it becomes increasingly difficult to patch clamp Drosophila muscles as temperatures increase. The recording electrode should have a final resistance of 5 to 10 megaohms and be heat polished to about 1 micrometer inner diameter at the tip. Recording electrodes are backfilled with a physiological solution containing potassium chloride and sucrose. A chloride silver ground wire is placed in the bath containing standard saline, which is also used in embryo dissection. Signals are amplified using a patch clamp amplifier, filtered and stored digitally for later analysis. Any system suitable for patch clamp experiments can be used, such as this equipment and software by Axon Instruments. Cells from 3 different tissues in the embryonic nervous system can now be stimulated and recorded from the muscles, neurons at the NMJ, and central motor neurons. With this embryo preparation, any embryonic muscle may be recorded from. Most experiments focus on the group of 4 longitudinal ventral muscles, 6, 7, 12, and 13, particularly muscle 6 in the anterior abdomen, A2 to A3. To record from the dorsal muscles, the embryo must be glued dorsal side down and dissected along the ventral midline. In young embryos, no enzymatic treatment is needed before muscle recording. However, in later embryos with developed cuticles, a muscle sheath also develops. Prior to patch recording, this sheath must be removed by brief immersion in a collagenase. Most recordings are done in the whole cell recording configuration, achieved easily from the cell-attached state with slight suction or an electrical buzz.
It's a zap. It's an electrostimulation to rupture the cell membrane to get a whole cell setting.
Seal resistances on the muscle are typically greater than 1 gigaohm. Whole cell currents are usually obtained less than 3 minutes after seal formation. Series resistance is markedly increased in most instances, and cells can develop an increased leakage current. For patch clamping, embryos must be at least 14 hours old. At earlier developmental stages, extensive electrical coupling between myotubes will make voltage clamping inaccurate. To make patch clamp recordings of muscle fibers, typical holding potentials are -60 to -80 millivolts, and voltage clamp and current clamp methods can be used. Successful whole cell recordings at -60 millivolts are accompanied by robust spontaneous synaptic currents that range from a few to several hundred picoamps, such as those shown here. Large NMJ currents can be evoked by stimulating the motor nerve with suction electrodes. These can be pulled from a variety of fiber-filled glass electrodes and should be fire polished to achieve the desired configuration. Using gentle suction on the pipette, draw in a small segment of the appropriate segmental motor nerve near the CNS exit point to form a tight seal. Brief stimulation works best with an experimentally determined intensity, usually between 1 to 5 volts. Reliable stimulation is achieved by setting the stimulus strength 1 to 2 volts above the experimentally determined threshold. Postsynaptic NMJ glutamate receptors can be activated by applying the neurotransmitter glutamate. Charged molecules like glutamate can be delivered to the NMJ using iontophoresis. High-resistance pipettes are used to map receptors, and lower-resistance pipettes are used to estimate the total glutamate receptor response. Iontophoretic pipettes are loaded with a 100 millimolar L-glutamate solution. To release the L-glutamate, use a short pulse of negative current with a small positive backing current to prevent leakage. Glutamate or any uncharged molecule can also be delivered to the NMJ using pressure ejection. For pressure ejection, use 1 to 2 millimolar glutamate and apply 5 to 10 psi of outward force to the pipette using a picospritzer. NMJ currents evoked by nerve stimulation or glutamate application at -60 millivolts are typically 1500 to 2000 picoamps. Many embryonic neurons can be individually identified based on soma position and projection morphology. Most experimental recordings have concentrated on a group of 5 superficial dorsal neurons within the embryonic ventral nerve cord. To record from these neurons, the ventral nerve cord neurolemma sheath must be removed with brief focal treatment with protease. The cell bodies are desheathed by delivering 0.5 to 1% protease in recording using a large diameter patch pipette. Suck the sheath material into the pipette containing the protease. Gentle mouth action can be used to repeatedly triturate the sheath in and out of the pipette. After 1 to 2 minutes, the sheath will rupture. The cells are now exposed for whole cell patch clamping. This recording paradigm does not require that saline be circulated or warmed. The external recording saline in this preparation is different from what is used in recording from muscles. The patch pipette can be loaded with lucifer yellow to later visualize the position and morphology of the patched cell. Here is a central motor neuron that has taken in lucifer yellow and visualized using fluorescence microscopy. Whole cell voltage clamp recordings are made at holding potentials of -60 millivolts at 18 degrees Celsius. Current clamp recordings are also possible. Neurons can be stimulated with the neurotransmitters acetylcholine or GABA. These are applied at the cell soma using iontophoresis or pressure ejection, as when recording from NMJs. Once a whole cell connection has been established, spontaneous currents are first observed. This is 1 minute of raw data obtained from embryonic ventral longitudinal muscle 6, patch clamped at -60 millivolts. In the absence of stimulation, a healthy preparation typically shows robust spontaneous synaptic activity, represented here by the many downward current deflections. Here is data from the same muscle with stimulation applied using a suction pipette on the motor nerve leading to that muscle. This resulted in multiple downward deflections of the current, representing evoked junctional currents, or EJCs. This is data from the same muscle stimulated by 1 millimolar glutamate applied using pressure ejection. The glutamate opens receptors, leading to large downward deflections in the current. 2 traces are shown representing 2 subsequent applications of glutamate.
I just shown you how to electrophysiological record from late-stage Drosophila embryos. Electrophysiological recording generally use standard patch clamping techniques, which we could not describe here in any depth. Ideally, anyone seeking to record from the Drosophila embryos will have already learned patch clamping using an easier preparation. That's it. Thanks for watching and good luck with your experiments.